Welcome, life science learners, to another installment. I trust that you guys are well. Today we're focusing on mutations and we're trying to understand what genetic profiling is. If we recollect, in the previous lessons we've looked at DNA, we've looked at protein synthesis. Today we're looking at genetic mutations. You might have heard of the term mutations. How do they occur? That's interesting. The significance of mutations, also very interesting. So let's try and understand the context of all of that in our lesson today. Very interesting question as we start off. And I'd like to pose this question to you all. Can mutations be helpful or harmful? So we often refer to mutations and we think, well, a mutation is harmful, it could be really damaging to you. Have we ever wondered whether mutations can be beneficial or helpful? And the other question is, do we have mutations? And I'll leave that question with you and I'll try probably attempt that at the end to answer that. So, as an overview, we're going to spend some time looking at some important terminology. We're going to try and unpack what a mutation is. Important to us is understanding the types of mutations, and we will spend some time in this segment looking at what is it that causes mutations. So some key words, again a recap of the word gene, and we know that genes are genetic sequences that code for characteristic. So if we talk about a mutation, we refer to a change in the gene sequence. So that's important to have context to the word gene. We also know that there's changes in the chromosomes of an organism, and so we're going to point to the process of a mutation happening on a chromosome. We need to unpack what a mutation is. We're going to look at factors that cause mutations and refer to those factors as mutagens. During our process of understanding genetic profiling, we're going to look at the concept of what coding DNA is and non-coding DNA. It's important for us to understand what DNA profiling is and its uses. And during that process of DNA profiling, we're going to refer to the abbreviation STRs and try and understand what short tandem repeat sequences are. We're going to look at the use of restriction enzymes in the process of DNA profiling. A concept that will be elaborated on during the lesson would be PCR, which is essentially a process of copying the DNA. And then we're going to look at the process of electrophoresis, which is using some form of electrical charge to separate out molecules. Okay, so let's get straight into trying to understand what is a mutation. A mutation is a permanent change in the DNA sequence of a gene. Let's try and understand that again. So it is a change in the sequence of a gene. Okay? Mutations in a gene sequence can alter the amino acid sequence of the protein encoded by the gene. Mutations can occur spontaneously, meaning very unpredicted, can happen at any time in all living organisms. However, we need to understand that not all mutations are inherited. Only those that occur in the sex organs during gametogenesis, which is a formation of gametes, can be inherited. So let's pause at this and try and understand the context of what mutations are. So as I mentioned, that mutations are a change in the sequence of a gene. And so if we think of what a gene is made up of a sequence of nucleotides coded by specific number of nitrogenous bases. These mutations mean that there has been an alteration or a change in that sequence. And so if we recollect that a gene codes for a characteristic, if we change a sequence of a nucleotide, it could in turn affect the mRNA that is produced. And so we say that that could in turn affect the mRNA sequence. And if you recollect, mRNAs contain codons, which are short 3 bases that code for an amino acid. So if there is a change in the nucleotide sequence of DNA, that could affect an amino acid sequence, and that in turn could have an effect on the protein that's being produced. And so we say that there's a change or an alteration of the genetic makeup. What is also important to recognize is that mutations occur spontaneously, meaning that these cannot be predicted. They can come up now and then without necessarily having some predictive nature around them. So they're spontaneous. What is also important is that we noted that mutations are inherited. That means they passed on from one generation to the next or from one cell to the next. And this can only happen if it occurs in what we refer to as the gametes. And those gametes are the sex cells, so it's the sperm cells and the egg cells. And that happens during the process of gametogenesis. Cool. So where and when do mutations occur? So as we said that mutations are spontaneous, they do occur very unpredictably. However, mutation is fundamentally a mistake. And these mistakes occur, one, when DNA is copied. And as a result of the environmental factors such as UV light or even using certain substances like smoking. Mutations can occur during DNA replication if errors are made and are not corrected in time. So guys, if we try and understand what DNA replication is a process of copying DNA. And if you go back to understanding that we spoke about different models of replication, where the DNA was copied, and one model that we refer to is the semi-conservative model, where at the end we have two new strands having half old and half new strands of DNA in them. And so because of the rate at which DNA replication occurs, we know that at some point mistakes do happen. The mechanism of DNA replication has a corrective process which corrects any mistakes that happen. However, some mistakes do go by without being picked up. And often when a mistake hasn't been corrected, we say that that alters the genetic information in the next cell or the next generation. And we say that that is now an inherited mutation which has not been corrected. So mutations may involve anything from a single base being changed to an entire chromosome. And that takes us to the next concept. Mutations can occur in somatic cells. So these are your cells in the body, excluding the sperms and the eggs. And this can occur during meiosis and mitosis. So let me rephrase that again. It can occur in mitosis as well as in meiosis. And this is during, as I mentioned, gametogenesis. Broadly speaking, there are two types of mutations. I refer to them as chromosomal mutations and gene mutations. Let's try and understand the difference between chromosomes and genes. Remember that in the nucleus we've got chromatin network, and that chromatin network consists of strands of DNA. When the cell prepares for cell division, it will arrange the genetic information or the DNA into compact structures called chromosomes. We also know that a chromosome consists of many genes. So on the DNA we have long segments of genetic information broken up into genes. So when we refer to a chromosome mutation, it's important that we recognize that that's a chromosome or that's a chromatid. When we refer to a gene mutation, it's important that we recognize that that's a sequence of DNA, and if a sequence of DNA has a single change at a specific point, then we refer to that as a change in the gene, referred to as a genetic mutation or a gene mutation. So let's try and unpack chromosomal mutations first. Let's try and understand what a chromosome is. So any change in the structure or the number of chromosomes is referred to as a chromosomal mutation. So if we have a chromosome, and if there's a change in that structure where a piece of that chromosome is broken up and lost, we can refer to that as a chromosomal abnormality or aberration. So there's a piece that has been lost. That's an abnormality that has occurred. That's a gene mutation. We know that some individuals may produce a gamete that has an extra set of chromosomes. And when that gamete fuses, we end up with an individual having a triploid set of chromosomes for a specific chromosome. So we refer to that as, again, a chromosomal abnormalities. It's important for us to recognize that chromosomal mutations occur at a large scale, which means that there are several genes that are affected. If we try and unpack this and refer to this chromosome, it consists of many genes. And so if we have a chromosomal mutation, it could affect more than one gene. And so we say that the effect is significantly larger because chromosomes contain many genes. Chromosomal mutations are sometimes referred to as chromosomal aberrations. And the word aberrations essentially points to the word mistakes or changes. So when we look at chromosomal mutations, we know that there are certain environmental factors such as radiation, ultraviolet, nuclear radiation, some chemicals, and even viruses that can cause chromosomal mutations or chromosomes to break. So we refer to that as environmental factors or mutagens. If the broken ends do not rejoin the same pattern, this will cause a change in the chromosomal structure, and we refer to this as a chromosomal aberration. There are several different types of chromosomal mutations. At this point, let's try and recognize what this means. A deletion of a chromosome would mean that a chromosome has been omitted from the gamete. We often see in certain conditions like Down syndrome, we see a duplication of a chromosome. So a Down syndrome individual will have, a normal individual has 2 sets of chromosomes for each pair. However, when we look at a Down syndrome individual, they would have a chromosome that has been in a triploid set. So it's trisomy, and that's because one of the gametes had a duplicate number of chromosomes, number 21. We sometimes see the inversion of a chromosome, which essentially is a segment of a chromosome. I'm going to use this illustration, breaking away and being inverted or swapped its position around. So we say that there's an inversion or the turning around of a segment of that chromosome. The word translocation refers to changing the position, and translocation essentially refers to when a chromosome arm breaks off and then moves and reattaches onto a different chromosome, and we see now an attachment of a different chromosome onto an unrelated paired chromosome. So we refer to that as transferring or changing the position of a chromosome. We also know that in some processes, like during meiosis, there is a process of non-disjunction, which essentially refers to the non-separation of chromosomes during anaphase. And so when we refer to anaphase 1 and 2, we know that these chromosomes are pulled apart, or we see the separation of chromatids apart. However, the term non-disjunction refers to when an entire pair of chromosome is pulled to the one pole, or when an entire chromosome is pulled to the one pole, as opposed to splitting up into chromatids and being pulled to the opposite ends. So we refer to that as non-disjunction. And we do recognize that this process creates chromosomal mutations. So as a wrap of this segment, let's try and review what chromosomal mutations are. So I mentioned that they could be the relocation of genetic material, and that can occur through translocation or inversion. And in this image, we're seeing a specific sequence where that's the gene, or if we kind of focus on an area of that segment, we're seeing that now we're seeing a segment from another chromosome being added on here, so that's changing its location. In this image, we're seeing that there's been a swap between that chromosome, in that chromosome where a segment of that chromosome has been changed in its position, which I've referred to in this diagram here. We sometimes see that there's a duplication of a segment of a chromosome, as you would see here. There are 2 sets of the original chromosome, so that's duplicated. or as I mentioned, in the case of a duplication of the entire chromosome, where we see two sets of one chromosome, which is different to the normal one set per gamete. Or we could see that there is a loss of genetic material where an entire chromosome has not separated, and you end up with a gamete that misses or lacks a specific chromosome. We also know that there could be a deletion of a segment of a chromosome, which we see absent in this specific chromosome. So guys, we've spent some time looking at the process of how chromosomal mutations take place. We're going to get back and we're going to spend some time looking at gene mutations and what happens during genetic mutations and the effects of those mutations. You guys have done well. You need a little break, stretch, come back in a bit, and we'll join you for a good interactive session looking at the effects of mutations.